I'm Riya Naik, the president of the Rocky Hill High School's research team. Today, I'm going to talk about bioethics presentation along with Sahana, Ganesh, and Neel Shah. We all are going to talk about bioethics because we all think that it is an important topic to discuss because we are experiencing a large amount of advancement in this field, and we think that discussing the limits and the boundaries is important while understanding what the advancements are. So here we go. What is biotechnology? It is the ability to edit the genetic code. Purposes can range from the treatment of any kind of pathology, modifying or enhancing traits in an embryo, or to the extremes causing fear or panic through bioterrorism. So the central question that we're going to ask today is, are the scientists playing God because they have the ability to edit the genome? Does that mean that they are the gods? So our beloved Spider-Man has said that with great power comes great responsibility. Yes, we do have the responsibility to act judiciously when we have the ability to edit the genome because it can cause fear and panic if not handled correctly. But at the same time, it has the great potential to help cure many diseases which we did not have cure for before this technology. So it is important that we consider the ethical issues before delving in this opportunity of editing the genome. So is genetic engineering ethical or not? What are the boundaries to edit the genome? Here we believe that the genetic engineering should be limited to the treatment of various pathologies and should not be applied for enhancing traits because it can cause social inequality if someone has the ability to edit the genome but there are many people who do not have the ability. So will that cause social inequality? That's a deep question that we need to ask ourselves. So the overview of this presentation today is bioterrorism, CRISPR, and epigenetics. So I invite you to this amazing tour led by these genius young scientists who are equally questioning the bioethical considerations while also appreciating this technology. So I hope you learned something new today, and I hope you understand the pressing questions before appreciating this technology. Thank you so much. Hello everyone, I am Riya Naik, the president of the Rocky Hill Research Club. Today I am going to talk to you about CRISPR and the ethical considerations while exploring this amazing biotechnology. So last summer I wrote a paper on the potential of the CRISPR, and this is the abstract from this paper. CRISPR, or clustered regularly interspaced short palindromic repeats, is based on the bacterial immune system. It was discovered in the 1970s in the E. coli bacteria. It is an anti-infection mechanism. If an analogy is to be drawn, the CRISPR-Cas9 system can be compared to the antibodies in the humans. It targets the viral DNA and creates strands of this targeted DNA when the bacteria is under attack from the virus. It creates CRISPR fragments and are stored as a memory when there is an invasion by the same virus. Similarly, in the lab, a biologist can target a specific sequence of the gene that is causing a mutation in the phenotype and can use the CRISPR scissors to cut the DNA at a specific site. It can be useful for cancer cures, disease prevention, and treatment to the genetic diseases. However, are we playing gods here by editing the genome of unborn children? That is the central question that we are asking today in this presentation on bioethics. It is a brilliant technique that has the potential to cure diseases, which were a death sentence earlier, the development of this technique. However, what are the boundaries? The ethical considerations are it can be CRISPR can be useful for treating diseases that affect a good lifestyle, like cancer, genetic disorders like cystic fibrosis. However, it may not be ethical to apply to enhance traits, as example, designer babies. social considerations are not all the children have the opportunity to have their IQs enhanced because this can be a pretty expensive technique, and only people who have means for this technique can enhance their traits. However, this can cause a social imbalance and cause children to have their IQ tests or SAT scores to be skewed. Also, we do not have the consent of the unborn child. Parents may not know if the child will appreciate the enhanced traits in the future. The child may have problems in the future and can suffer consequences of their genes being edited when they did not have the understanding of it. So we do not have the consent of the unborn child. So to discuss these questions, the United States National Academies of Science, Engineering, and Medicine had invited the Chinese Academy of Science and the United Kingdom's Royal Society in 2015 to discuss the ethical guidelines for CRISPR. This committee decided that somatic editing is permissible for the cure of genetic diseases. However, the purpose of the enhancement is not permissible. To date, these guidelines are maintained as embryo research still remains controversial. So you can access my paper on CRISPR on this link. I will add in my bio on the YouTube channel. So I hope that you enjoyed this presentation today with about CRISPR, and I hope you understood some of the limitations and how this technology works. Thank you and have a good day.
Hey guys, my name is Neel Shah, and I will be going over how genetic engineering is revolutionizing bioterrorism. So just going through my abstract, genetic engineering technologies have been well suited in the medical field, creating advances in the production and delivery of drugs and other forms of medication. However, there is a catch. Attention towards biowarfare have remained quiet in many nations until genetic engineering turned the tide. It may be possible to fabricate diseases or other harmful pathogens with no fail. In this presentation, we are going to look at how genetic engineering brings some unsettling prospects into creating bioweapons and its other ethical implications. So, what is bioterrorism and biowarfare? Well, essentially, it's a deliberate attack through delivering harmful biological agents out into the world. And examples of these biological agents would include bacteria and viruses. But before genetic engineering, there were natural pathogens found in nature transformed into bioweapons. And now labs can fabricate these bacteria and these viruses, which we'll get into later. And who creates and deliver these bioweapons? They would be terrorist organizations, but also under biowarfare programs, sometimes even managed by the government, which we'll also take a look at in the next slide. So, what are three key parts in effective biowarfare? One is finding suitable virulent agents, which basically means that these organizations would pinpoint characteristics of preference based on their genotype or their genetic characteristic for their virulent agents, such as a gene that can target a certain ethnic group or a certain other group. And another key aspect is mass production without losing pathogenicity. And that means when the virus is mass produced or replicating or dividing in this case, it can easily lose its virulence or its ability to be viral and spread and affect people. And lastly, the hardest part is effective means of delivery and configuring that whole aspect of delivering it out into the world without failing. So now getting to some background, when I was talking about the biowarfare programs, a key underground biowarfare program would be the Soviet Union in the 20th century, and that basically led to their notorious anthrax attack, which basically was introducing an alien gene into Bacillus anthracis, which altered the immunological properties and thus making known vaccines and treatments ineffective to the strain of anthrax. And because of that, many, many people died. In the 1925 Geneva Convention and the 1972 Biological and Toxin Weapons Convention, it was actually realized a lot of countries have been using offensive or taking advantage of offensive biowarfare research. So at that point, many countries stopped using bioweapons because it was unethical, such as the US in 1969. But now in 2002, researchers at the State University of New York, Stony Brook, actually synthesized an artificial polio virus. And that's dangerous because it can prove that with the technology right now in the 21st century, that labs also have the capabilities of fabricating a deadly virus. And if in the wrong hands, it can be chemically brought to life and also spread as a potential bioweapon even. So we're gonna talk about the genetic engineering and the Human Genome Project and what impact this really had. So basically, genetic engineering is transferring genetic material between species, and basically through technologies such as CRISPR, gene electrophoresis, designer genes. Um, Basically, these technologies have the ability to edit genes and even change the characteristics of some organisms. And the Human Genome Project was a 13-year-long project from 1990 to 2003, where researchers all over the world sequenced human genome. And the Human Genome Diversity Project in particular um, basically sought to find information that could be um, that basically like figures out the genetic differences between ethnic and social and health groups. And this information could be used, like I said before, to target weapons at certain groups, most notably ethnic groups. So, talking about the before picture, um, basically, I talked about the prerequisites of effective bioweapons before, the three key aspects. And just talking about that, there were many binding limits before genetic engineering was producing bioweapons. It was even harder because all the pathogens were natural, so it couldn't really be altered. There wasn't a huger extent of it being altered. The labs had to work with what they had. And it was harder to produce in larger quantities because they needed to have enough of these pathogens to test its qualities to figure out if that was like the pathogen that they really wanted. And then these pathogens could also be very hazardous in the lab environments. Then it could be also susceptible to losing its pathogenicity because they need to divide and divide and they need to be produced. And obviously, with genetic engineering, it was impossible to alter or even fabricate new pathogens. So it was hard because of a lot of natural pathogens weren't really accessible. But now with genetic engineering, bioterrorism could actually be more feasible because even though people were not really nations who previously banned bioweapons before are now taking another look at it because of genetic engineering. So now that we have technologies such as DNA synthesis and DNA sequencing, we can actually create novel pathogens, such as a synthesized poliovirus. Synthetic diseases are more effective than their natural counterparts at this point because now we can, pick and choose which properties we want to put, like antibiotic resistance, so that it can be immune to antibiotics and other treatments. And now it's very, very more feasible than the anthrax attack. And even you implementing antibiotic resistance and having this could also lead to, for example, if a nation develops a bioweapon and they disseminate it, they can also, since it's their own creation, they could also create a separate treatment that, in the case that they're like soldiers or if any of their researchers get infected, then they can treat them, but they won't treat anybody else, specifically their target. And now, um, since like I said before, many nations ban bioweapons, and now they're taking a second look. So that's frightening. And with high tech facilities too, since labs have many more capabilities now than before, they can easily produce these strains. So the benefits of genetically engineered pathogens will basically be the easy access to information. Now that all these genomic information, they're all in the public domain. There are a bunch of genetic databases out to the public. And bio warfare programs who produce these bioweapons can easily go undetected, since the whole most of the purpose of genetic engineering, most of these purposes are already towards medical science. So many of these programs could adopt that same supposed like purpose, but then actually go undetected and actually produce bioweapons. And now, actually, these are much more easier to produce in larger quantities since they can easily maintain its virulence, and these agents are also easily delivered as well. So the potential methodologies, I'm just going to explain a few. So we have binary biological weapons, which basically would be implementing plasmids, which would contain like the gene of interest or like the genetic characteristic into other bacteria so that it could, for example, enhance the virulence of the bioweapon or alter its other pathogenetic properties to the advantage of the creator. And other supposed methodologies would be stealth viruses, which basically would be viral infections that enter host cells and actually remain dormant until it's triggered through a specific disease. And in the context of biowarfare, triggering this virus can be used as a form of blackmail. And at this point, we can personalize bioweapons too, because now like biotechnology is so expansive. Um, Basically, pathogens can honestly be used to target a specific person's genome with gene sequencing technology. So now we're going to get into biodefenses. There are a couple. The human genome literacy, for example, basically is a database which identifies the functions of certain genes, and they may be how they are used to cause disease. So with this vital information, then we can produce vaccines and drugs towards a particular disease on a molecular level with that information. And then there's also immune system enhancement, which also is permitted through the human genome literacy because we also have more in-depth information and understanding of our immune system. So genetic engineers can try to bolster the human immune response to pathogens. And the viral and bacterial genome literacy allows us to have more knowledge about the bacterial viruses and also give it insight to their virulence and drug resistance. And um, because of that, bacteria can also be engineered to produce bioregulators against these pathogens that we know. So now the ethical implications. Obviously, it's mostly going to be negative because, with the exception of bolstering biodefenses, because obviously bioterrorism is not a good sign. So a lot of scientists are speculating that the third world war with these new technologies will be predominantly biological, and this could also lead to the potential of a biological arms race. And the like I said before, genetically engineered pathogens are attracting many nations, and that can also mean that they're attracting terrorist organizations as well. And even though we have like a bunch of these genomic databases which can be used to our advantage, it could also be used to our disadvantage as well. Pathogens are likely to be produced that are resistant to certain biodefenses that we already know and that are already out there. And military desires can also be shifted because of this widespread attention to genetic engineering. And last but not least, there can honestly, with how expensive biotechnology is, then there can even be a super bug that can be created. There is technology to create it. So it's pretty unsettling, but um, with this information, I hope you guys get a better idea of how genetic engineering is um, unethical on the side of bioterrorism, because I feel like this is not really as well known, bioweapons in general. So hopefully this gives you a lot of insight into how biotechnology can also be taken advantage of and abused.
Hi, I'm Sahana, co-leader of the Rock Hill Research Club, and I'll be introducing the topic of epigenetics to you all. To understand this topic in biology, let's first understand its background through an abstract. Humans have long known about the double helix structure inside every one of our millions of cells that shapes our personality, our beliefs, appearance, and much more. So far, this structure has been thought to be set in stone, with beliefs that we cannot change who we are genetically. However, epigenetics brings the idea that our environment can shape who we are genetically, contradicting our held beliefs. The theory that we could tinker with DNA and produce different, interesting results to possibly cure life-threatening diseases and make life a whole lot easier has brought on many endeavors to complete this physically in a digital world. However, there are plenty of ethical issues humans have to face. even if we had the right technology to do it. This presentation on epigenetics will help people understand the ethicality that comes with decisions made in science, as well as introduce new topics that change the face of research and future endeavors. Let's first define what epigenetics really is. It is a study of the changes in organisms caused by the modification of gene expression rather than changing the actual genetic code itself. The cell only reads its specific genes that are expressed, which then is expressed through phenotype, which are observable traits like eye color or hair color, rather than your actual DNA, which is your genotype. An easy example of this is through the development of stem cells, which are cells with the potential to develop into many different types of cells in the body. Let's say there are two stem cells. Both of these stem cells have the same DNA. However, stem cell 1 develops into an ear cell, while stem cell 2 develops into a lip cell. Why does this happen? It's because different genes are being expressed, which then changes the physical and observable traits that we see. Therefore, it is not that the DNA is being changed that causes two similar cells to become two different things. It is through what specific genes are being turned off or on that determines what happens physically. Overall, epigenetics basically decides how much or whether genes are expressed in your body. Now let's talk about why these epigenetics changes happen and what are some impacts. There are many causes of epigenetic changes, biologically and environmentally. Biologically, methyl groups in the body, which are basically one carbon atom bonded to three hydrogen atoms represented chemically by the formula CH3. These methyl groups play a role in DNA methylation, a process in which methyl groups are added to a DNA molecule. When this happens, the methyl group does not necessarily change the DNA sequence itself, but rather acts to suppress gene transcription, the first step in gene expression, in which an RNA polymerase molecule uses a DNA template to create an RNA strand. Because the methyl groups causes nucleosomes to pack tightly together, genes cannot be accessed and therefore will not be expressed. Histones is also a factor in gene expression and epigenetic changes. If DNA is more tightly wound around them, genes are expressed less as it is harder to access. If it's more loosely wound, they are expressed more because there's more accessibility. The age of the individual can also cause epigenetic changes, as the genes being expressed may change as the aging process starts and tissues get older. There's also a higher chance for diseases as the body gets more vulnerable. The environment of the individual is also found to be a great factor of epigenetic changes, since where that person is determines their type of lifestyle. For example, as seen in the picture, the kid who has access to a better nutrition, a good environment, and responsibility will grow up to be healthier than one who does not. Changes can also be hereditary or passed on from generations, which, along with other factors, brings about some major ethical disputes and conflicts to think about. Scientists have long pondered, disputed, and argued for what affects our genes. Is it strictly on our nature or DNA that determines our behavior? And we can never change who we are, essentially. Or can nurture, which relates to our environment, upbringing, and our own life experiences, affect our genes and our behavior as a result? Well, epigenetics completely refutes this whole debate and says that both our nature and our nurture affect our behavior and personalities. Essentially, both our genetics and environment can affect gene expression in our body. Some ethical implications of this is that individuals who have different living conditions, financial stability, environment, and so on have great inequalities that can deeply affect their behavior. It also affects the ethicality of pregnancy because of the fact that epigenetics is hereditary. Do parents have an obligation to keep their epigenome healthy as possible to make sure that future generations can stay healthy as well? Should people with life-threatening diseases and compromised genomes have children and the risk that it might pass down onto them? These are all ethical issues that scientists and even students should discuss as technology and new scientific techniques are being created to one day execute these ideas. To learn more about epigenetics, there have been many research experiments done throughout the years. Since we know that epimutations can be reversed, diseased cells, because of epimutations, can have their epigenetic processes reversed, which is more specific to cells than classic radiation or chemotherapy, which can also attack healthy cells with the compromised cells. At the present, these epigenetic therapies have been approved by the FDA, but we have yet to execute these techniques in experiments. These epigenetic therapies would involve reversing chemical tags attached to DNA that promote life-threatening diseases in the hope that the body would not have that disease anymore. These research have also been done in the past, specifically in northern Sweden in the 1900s, in which nutrition analysis noticed that older generations who suddenly had experienced an overwhelming high food supply after a famine became obese and died earlier than the generations in famine. This lower lifespan and obesity also transmitted to future generations, proving the hereditary characteristic of epigenetics. That is it for the presentation. I hope you guys learned some interesting information about epigenetics and the ethicality of such groundbreaking decisions to go inside our genome and change our God given characteristics.